Okay, hi everyone. Thanks for coming into the online lecture, and I just wanted to touch base with you really quickly because we're still on the topic of LTP. And just remember, and thanks for the memes. Big shout out to Zara Ahmed for sharing all of these with the class. I'm hoping that you are actually getting ready for the final exam and that you are spending some time every week getting ready for what is going to come up in December, because that's only 2.5 weeks away. So just as sort of a reminder, we're looking at learning and memory, and we're looking at some of the different contexts of learning and memory, and we had sort of left off looking at some of the basic ways in which learning and memory are normally studied. And I wanted to tell you in advance that today, some of the different topics that we're going to be looking at are actually quite heavy in terms of the content and what you need to know and how you're supposed to be able to predict different types of things that might be coming up. So we'll be talking again a little bit more about that in a few moments. So let me start off with a couple of different stories related to learning as well as memory. Did you know, for example, that emotion plays a huge role in learning and memory? And we already knew that that was the case because in the last lecture we had sort of talked about the different experiments that were done by Sandy et al., and I'll show that slide in the next figure. But in terms of emotion, in the Middle Ages in Europe, because there was no way to record things, for a village to actually keep a memory or a record of something that had occurred, a big momentous occasion, they used to take a 7-year-old boy, not quite the 7-year-old boy that you see here, from the village who had witnessed everything that occurred, whatever that was, the festival or some sort of celebration or something that was monumental to the progression of that village. And then in order to charge with what was happening with that particular event, they would throw them into the lake, a nearby lake, and because the 7-year-old wouldn't actually know how to swim, they would be struggling, and they would wait until the last possible second before they started to actually drown, before they would bring the boy out. And again, because of the trauma that was associated with it and the emotions that were encapsulated in that particular type of memory, it usually ended up being remembered for a very, very long period of time and throughout the time of that particular young individual. I also wanted to tell you a little bit about memory in general. So when you think back to something, whatever you think is a really important memory, how do you actually know that memory is true? And that's sort of what we're going to be looking at here today. How does memory get stored? How do you know that memory is true? So one of the things that I can tell you as a neurobiologist, and you'll have to believe me that I'm somewhat of a good neurobiologist, when I got married to my wife several years ago, one of the things that I remember very, very vividly is walking along a beach, much like this one here, waiting for the sun to go down, much like you see in this particular picture, and I was walking side by side with her. I can still feel the sand between my toes. I can still feel the warmth of the sun as it's still going down, and I can actually smell, even now, sitting here doing this lecture, I can still smell the salt air as I looked over at the sunset, put my arm around my wife, got down on my knee and proposed to her and asked her to marry me. And you might think, wow, what a fantastic memory, or what a great recollection of that memory. However, my wife and I are not sure, because my wife has a very different memory of how I proposed to her, that I had taken her out to a local restaurant, that we had sat down for dinner, and we had waited until dinner was served, and we were just waiting for the bill to arrive before I proposed to her. Which of those memories is correct? We'll never know. Always believe your professor, I guess, or if you're living in my household, always believe that my wife is correct. But that is a type of memory that we'll never really know the true story behind. So this goes back into what we talked about in the last lecture. We had sort of talked about the fact that different forms of memory under different types of stresses can actually either plateau, so emotion-laden, particularly very, very stressful and very anxiety-driven forms of memory actually plateau. They get stronger, versus other forms of memory, the types of memory that you need to function really well on your term test, these are saturable. And in fact, at a certain point in time, the amount of stress that you perceive will actually degrade the amount of memory or the type of learning that you can actually engage in. And you become less effective as a learner if the stress gets very, very high. And again, we measure things like stress and the levels of cortisol that we can detect within the blood. So today, there are a lot of different things that we're going to be going over. I will tell you in advance for both the types of things that we're going to be looking at on your quiz as well as for the final exam, that in particular I'd like you to make sure that you understand some of the different markers that we use for some of these different neuronal compartments, and we'll get into that in a few moments. I am going to tell you a little bit about one of the very first types of memory molecules, which is known as protein kinase A, and then we'll be looking at the hippocampus and how we can develop autopsies from these different types of hippocampal cultures. And in particular, we left off with CAM kinase 2, but we're going to be looking at different types of molecules today. So today we're going to start by going through the mind of the Nobel laureate Eric Kandel, why we study memory the way that we do, and why we think molecules like CAM kinase 2 are so important, but maybe not important for very long-term forms of memory. When you think back to the name of your 6th grade teacher, why is it that you can still remember your 6th grade teacher's name, even though you might not have thought of them for over a decade? We're not continually reusing that particular piece of information, but somehow that memory is encoded, and it probably doesn't involve CAM kinase 2 alpha. So we'll start looking at different model systems, why these different model systems are in use, and then this big race to find this molecule that's related to memory, and how the Nobel Prize laureate actually set off a rush to find out different aspects of learning as well as memory, and how we've actually gone full circle in terms of what we think learning and memory is involved in. So again, just for a little bit of fun related to our lecture, if you were to go and look at these 3 different individuals, which of these different individuals do you think, for example, is really important in terms of neuroscience and learning and memory? Hopefully you didn't pick B, because that's someone that we all know is a mediocre, I guess, rap artist, but we'll talk first about C, James C. Kirk. So if you picked James C. Kirk, that's not correct. And again, Aubrey, not really that important for neuroscience, maybe not really all that important for rap either, but we'll get back to that maybe at some other point in time. And then the person that we do want to talk about today, James McConnell. Now, James McConnell is this really controversial figure within the field of neuroscience, and you'll understand why James McConnell is a controversial figure as we go through a little bit more of today's talk. So when we're talking about different model organisms, we need to understand a couple of different things. So if we look at these different organisms, there are 2 that are shown for you here. This one here, the paramecium, is not one that we normally study in neuroscience. I just put it up there because I wanted to see if you knew what a paramecium was. And if you take a look over here, what do you think that this molecule is? And again, just keeping in mind, on your final exam, you will not have something like something else. So is it an anchovy? Is it a baby alien from the movies? Is it a C. elegans, or is it something else? And it turns out that this is something else. And we're going to talk about that something else in a few moments. So let's go all the way back to the late 1950s and early 1960s. This is a wild period in terms of neuroscience. In 1959, a Swedish neurochemist by the name of Holger Heiden is starting to think a little bit about learning and memory. How is it that we remember the name of our 6th grade teacher? What are the molecules that are involved in memory? And this was sort of the very first attempt to understand how memories are encoded at these synapses. Is it really just the strength of these AMPA receptors that's changing, or is there really a memory molecule that every time we think of something, these molecules have to be reactivated, and we have to get more of these molecules that are associated with that memory? So in 1959, Holger Heiden proposes that RNA is this molecule. So remember, by this time, DNA had been elucidated, RNA was found, and proteins had been known for a very long period of time. And because they are capable of being really rapidly modified, and because memory has to be rapidly modified, Holger Heiden suggests that RNA is this memory molecule, that the way we remember things is by changing the order of the sequences of bases found in RNA very, very quickly. So his proposal was RNA is sitting there, it gets reshuffled around by a memory, and that is how we remember something, because that RNA now has a completely new sequence. So he tests this out in 1962. So he's working with another neurobiologist, Egihazy, and what they were doing was to train rats to climb a wire in order to earn a food reward. And because, again, they wanted to find an area of the brain that they would think was associated with this form of learning as well as memory, what they found, or what they proposed, was that the vestibular nucleus, which is normally thought to be important for balance as well as movement, they decided to take out the vestibular nucleus after these rats had learned this particular task, and they purified the RNA, and what they found after learning had occurred in the vestibular nucleus was that they had an increase in the levels of RNA, and more importantly, even back in the 1960s, when they looked at the RNA itself, that there seemed to be a difference in the base composition. The overall nucleotides that they were finding had changed before training and after training. And it suggested that memory, the memory of being able to climb this wire, was actually stored within the vestibular nucleus by altering the RNA molecules, by changing the base pair pattern. Again, in the 1960s, after reading about this, James McConnell, the individual that I had shown you earlier, decided to test this hypothesis by using what is known as a planarian model. So that something else that I showed you a little bit earlier on tends to be this planarian, and this is an example of a planarian that you see swimming around. These are flatworms, and he decided to use these as his model organism in terms of learning and memory. So Holger Heiden and Egihazy decided to use rats. James McConnell decided that he wanted to use the planarian or flatworm to test this RNA hypothesis of being able to store memory. So again, you have to be able to teach the planarian something. It has to be able to learn this, and it has to be able to remember this in terms of all of these different molecules being involved. So James McConnell knew that planarians could move toward a particular food reward depending on where it was. So if he wanted the animal to move to the left, he would consistently place a food reward over on the left side of the particular swim tank. And if he wanted it to move to the right, he would do the same thing. If he wanted it to move to the left, he would actually shine light on the right quadrant. It wants to avoid the light, and it will move in the opposite direction. It remembers all of these different things, and so he could train it to move either to the left or to the right. So interestingly, he decided that after training, he would grind up the planarians that had just been trained. So they had learned to move to the left, or they had learned to move to the right. And then he had taken out the planarians, ground up all of the planarian, and then fed the planarians to naive planarians. So basically, he was making them into cannibals. And then he wanted to know, these naive planarians that had never been trained before. So imagine walking into an exam, eating the brain of someone else who had learned all the material, and then being able to sit for the exam and getting all the answers perfectly. And what he found was that the planarians that were naive, that had never learned this particular task before, actually moved in exactly the same manner as the trained planarians. This was mind-blowing. James McConnell had basically shown that you don't have to learn anything new, that simply by absorbing this molecule, whatever it was, and his hypothesis was, just like Egihazy and Holger Heiden, that it was RNA. And he thought that maybe this was the molecule that was involved, and he showed very clearly in his initial experiments that doing so, after training, grinding up planarians was enough to cause naive planarians to learn that type of behavior. So there was a lot of controversy about this, because when scientists tried to reproduce his experiment, many of them could not do so, and it turned out that he had not switched the plates in between, and they had left the chemical signal. The trained planarians had left the chemical signal so that the naive ones as they were going and swimming on these different types of plates that you see here, were simply following the chemical trail that had been left over by the trained planarians during their earlier attempts. Again, this threw back science and the whole area of trying to understand the memory molecule and trying to find the memory molecule, because basically scientists were thrown into a lot of disrepute about this whole field, that this was so controversial that all of the material that James Connell had published was under so much scrutiny that no one actually decided to pursue this consistently for a very long period of time until Eric Kandel. And Eric Kandel eventually won the Nobel Prize in 2000 in physiology or medicine for elucidating the molecules that underlied learning as well as memory. And in order for him to do so, he decided to ask some foundational questions. He wanted to know, at the level of the brain, what are the different circuits or what are the different cells that are involved in learning as well as memory? And he also needed to know where in the brain is learning and memory being stored. Where is this at an organizational level? He also wanted to know, how is this memory stored and how is this memory going to be retrieved, and what are the processes that are involved in interfering with this learning and memory as well as enhancing this learning and memory. So if I found a molecule and I introduced this molecule, would I be able to enhance learning and memory without actually having the organism undergo any particular type of training? So this goes back into first principles. So Eric Kandel wanted to find some sort of circuit that he would be able to understand. And this is always the case within science. Within the human brain, for example, our neural circuits, the nodes, and how the various axons connect different regions of the brain actually get very, very complicated. And so oftentimes we try to simplify things the best that we can. And often we go back to first principles. And one of the first things that we need to do is to develop a simplified model organism that has a nervous system, that has the capacity for us to be able to go in to examine this circuit at the cellular level. And oftentimes this means, because it's a simplified model organism, that the types of learning that are involved are much simpler forms of learning than we would have in a mouse or a rat or even in a human. So these simplified systems and these simplified forms of learning often involve things like invertebrates. It could involve simplified mammalian preparations, and I'm going to show you an example of this in the next slide, where, for example, to see synaptic strengthening, which is a little different than what Eric Kandel is looking for in terms of synaptic strengthening, that in fact these neuronal cultures are often much more simplified, where they develop what are known as autopsies. And so I'm going to introduce autoptic cultures for you in the next slide. So many of you will have done neuronal cell culture, and again, if you have or you haven't, it's really not that important. We can take individual neurons and then dissociate them, break them apart, and then grow them on a two-dimensional plate, as you see here, and we're looking at them under a microscope, and we can look at them 7 days in vitro. So if I plate them on day 0 and I look at them 7 days later, I say that it's 7 days in vitro, or I can look 21 days after initial plating, and you can see that they're very, very different. So here we don't have a lot of connections or a lot of axons that connect or dendrites that connect these different neurons, and these represent the neuronal cell bodies. But you can see that after 21 days, all of these dark lines represent the different dendrites and the connections between them. And oftentimes this means that when we take these dissociated neuronal cultures, we take them from embryonic brains, particularly from the regions of brains that we are interested in. So for example, we might be interested in taking out hippocampal neuronal cultures, and then we plate them on specially coated plastic or glass cover slips so that we can see them under a microscope, similar to what we are seeing here. They do require special growth medium in order for these neurons to be able to mature. And again, these days in vitro actually reflect the level of developmental maturity. So the ones that we see here are very immature neuronal cultures, and 21 days in vitro, they are much more mature. We see more synaptic connections. We see things that look more developed, similar to what we would see in an older animal. And again, these neurons are living cultures. But here, even though we have dissociated and can look at these individual synaptic connections, one of the things that we can do is to simplify this even more. So we have many, many different synaptic connections between these different groups of neurons. What if we simplified these dissociated cultures to an even simpler level? And one of the things that we can do is to generate what is known as an autopsy. Now, we do everything just like we did for a dissociated neuronal culture, but here we actually have very special conditions for plating. We have to have this specialized stamp so that we would actually press down into the medium. So we get these little islands where we can grow these neurons, and these neurons are grown in very, very low density. That means we only get one neuron in a field that we're looking at here. So this neuron cell body, the axon wraps around, and it is making its own synaptic connections with its own dendrites and its own axons. As a result, we call this an autopsy. And by doing so, we can then look within this autopsy to see what is the structure in this very, very simplified system of the different types of synaptic proteins that we can identify. And again, this is a good slide for you to make sure that you know prior to the quiz on Thursday, as well as for the final exam. So one of the things that we can do is to use a marker like proline-rich synapse-associated protein 1, or ProSAP1, and this is specific to the postsynaptic compartment. And then we can also go in and look at the presynaptic marker. So even though this is an autopsy here, VGLUT1 is a presynaptic marker, and it's particularly important in excitatory neurons because this is a vesicular glutamate transporter protein known as VGLUT1, and this is a presynaptic marker protein that we would find in the presynaptic compartments. We can also look at MAP2, which we've seen before, which is a dendritic marker, and we can see that even though it is an autopsy, all of the things that we would expect to see in a mature synapse are present in this singular system. So it's a simplified model, but we see that we have presynaptic elements and we have postsynaptic elements, and we also have dendrites. And it is making connections with itself, which makes it a very simplified system. If we were looking at synaptic strengthening, for example, we could use this autoptic culture to examine individual synapses on a neuron that makes connections to itself. However, although these were available to Eric Kandel, he decided that he did not want to use these autopsies, nor did he want to use dissociated cultures. He wanted to be able to look at behavior and to link this back to the neuronal circuit that he was able to identify. And again, when we look at these invertebrates in general, these nervous systems have some advantages. They have a much smaller nervous system. They don't have billions of neuronal connections or trillions. They have a several orders of magnitude lower. These neurons generally are much, much larger, making it easier to do electrophysiological experiments. We can also identify each of these neurons within the circuit because we can see them and we can record from them. And this means that these identifiable circuits could be potentially involved in a type of behavior that we can go back and examine. Also, a lot of these invertebrates, C. elegans is an example of an invertebrate, have much simpler genetics, and we can go back and look at the genes that are found within these identifiable neurons that make up these different types of circuits. And again, although it was available to Eric Kandel, he decided not to use this particular C. elegans model either, but all of these different characteristics and these different features are also true of the organism that he decided to use, which was the Plisia californica, which is what you were seeing here. This is a giant sea snail. And he decided to use this to unravel some of the synaptic mechanisms that underlie learning as well as memory. And he basically set up the template that we still use to this day in 2018 to identify different molecules that are important in synaptic plasticity, the underlying behaviors that are involved in these different circuits that we have been discussing up until this point in time. And of course, he did win the 2000 Nobel Prize in Physiology and Medicine for this work. This is not a slide that you need to spend a lot of time memorizing, but one of the things that he was able to do, going back to what we know, the aplysia is an invertebrate, so it does have a very simplified nervous system, and these neurons tend to be very, very large, allowing for electrodes to be easily implanted within these different groups of neurons. Each of these, in turn, have been identified, and we can identify each of the behaviors that are associated with them. So for example, one of the things that he was able to show is that there is a sensory neuron that makes connections to the skin, and that if he either shocked the skin or he squirted water on the skin, the sensory neuron would fire. It would then activate this motor neuron, which he is able to record from, this L7 motor neuron, and that this would cause the siphon gill muscle to retract. So by shocking the siphon directly, he was able to cause the muscle to contract, which is normally what is observed in the sea snail itself. So in response to stimuli, it will actually learn to retract the muscle, and the siphon is retracted as a result. And as it's learned this, whenever it encounters a squirt of water, it will retract that siphon that it has learned to associate with the shock for prolonged periods of time. So we can determine how long this particular form of memory can exist for. So one of the things that he found was in this system, the activation of protein kinase A was essential. So he showed basically, similar to what we now see in the hippocampus, that a kinase is important in initiating all of these different forms of activity. So one of the reasons why we're so interested in CAM kinase 2 is a direct legacy of what Kandel had found. So what we are left with in terms of Kandel is that we have this neural basis of memory from these invertebrate studies, that learning and memory can result from modifications. So he showed that this synapse here became strengthened because of protein kinase A. So as it learned this association, the sensory neuron would fire more frequently. It would increase the levels of protein kinase A, allowing for this gill withdrawal reflex to occur. So this involved the modification in that synapse, and that this involves second messenger systems, ultimately resulting in protein kinase A. And as a result, these different types of synaptic proteins would also be modified at that synapse, which is what we look at now in terms of AMPA receptors and how they get modified at the level of the synapse. So this is basically the blueprint that we do now in terms of learning as well as memory. And this goes back to what was originally hypothesized by Donald Hebb, that Hebb had hypothesized that memory results from this synaptic modification. This, again, also results in what we still do to this day. We try to simplify these different model systems as much as we can, and we can also look at how we can directly stimulate these different fibers, and we still do that in terms of high-frequency stimulation that we've looked at in previous lectures. So again, when we look at some of the rules that are associated with LTP, there are a lot of rules that are in common. We know that only some synapses become potentiated. We also know that neurons have a discrete level. And at least up until 2016, this was true. And in 2018, some of these new rules now suggest that maybe some of these synapses in certain areas of the brain can actually continue to be strengthened, but this again remains to be very, very controversial. We have to have a presynaptic element and we have to have a postsynaptic element, and we think that most of the information is encoded at the level of the postsynaptic proteins that are getting altered. Again, we think that the hippocampus is important, particularly in the model systems that we've been looking at, and we know a lot of this information comes from HM or Henry Molaison, but we know that the hippocampus itself is not where we store long-term memories, and I sort of left things off at the end of the last lecture with these remaining slides. We know that the hippocampus is important for retaining memories for several weeks before it actually gets stored or consolidated elsewhere within the cortex. So LTP is really a short-term form of memory within the hippocampus, but it really isn't like long-term memory. When you're thinking about your grade 6 teacher, so more than 10 years ago, that's really not LTP. You're not strengthening that synapse. That's a pure memory that has been stored and encoded and consolidated elsewhere within your cortex. And this hippocampal form of LTP that we looked at and what Eric Kandel is looking at in the aplysia is very different than what we're going to be looking at for the remainder of today's lecture. So for these stable long-term memories, we have to be able to basically mirror the strengthened synapses where we get AMPA receptors being strengthened at the level of the cell surface of these CA1 hippocampal neurons, and we have to be able to mirror this within the cortex. So somehow these strengthened synapses, the signals from these strengthened synapses have to leave the hippocampus, and the pattern of those synapses have to leave the hippocampus via axons of the pyramidal cells that will eventually form what is known as the fimbria fornix. So we had learned about the entorhinal cortex and how information comes into the hippocampus, but now we're looking at how the fimbria fornix causes information to leave the hippocampus, and the axons of the CA1 pyramidal cells will eventually form this structure known as the fimbria fornix. Again, the hippocampus is involved in short-term memories, the ones that you are generating over the short term, over the course of like 1 hour or the course of several hours, but for it to be stored in long-term memory where you're going to be able to access it over years at a time, it actually has to leave through this fimbria fornix. As you would imagine, lesions to the fimbria fornix therefore also lead to deficits in declarative memory, and again, it becomes a very interesting sort of field to be able to go in and study. So somehow we have to get past this notion of LTP, which is what we talked about in the last lecture. So the last lecture, your hippocampus is trying to strengthen some of its synapses. It is going to be involved in long-term potentiation, which is going to be stored in the hippocampus, and this will be a short-term memory that might last for an hour, or it might last for a period of a couple of days, but beyond that, we need to move that information, those strengthened synapses, those AMPA receptors, all of that has to be mirrored and has to be recapitulated at the level of other synapses within the cortex. And what are the molecules, therefore, at the level of the cortex that we have to alter inside the cortex? So this, again, is time dependent. We need to be able to transfer this memory. You need to be able to figure out a way to remove this memory from the hippocampus into other areas of the brain. And we need to also get this strengthening of these synapses, and we're particularly talking about LTP, but we don't really know if this also applies to LTD in different areas of the cortex. And typically, again, we're going to be talking about glutamatergic synapses for the most part, but we don't know if this is also true for inhibitory synapses or for every type of synapse that we might be able to examine. So again, in terms of the hippocampal circuit, just as a review, we have been talking about integrated sensory signals, vision, sound coming in to the hippocampus through the entorhinal cortex. Through the perforant pathway, it will send their axons into the neurons within the dentate gyrus, which then send information into the CA1 area of the hippocampus. These CA1 axons in turn will send their axons out, and collectively these axons are known as the fimbria fornix, and these will leave in terms of the hippocampus out into the cortex. I sort of left with this slide because now we're kind of moving in beyond LTP. We're now looking at what happens after the hippocampus. What happens when we actually start developing long-term memories. And this becomes a very, very controversial field in the area of neuroscience. And this gets into, who do you believe is actually correct? Do you believe Todd Sachter over here on the left, or do you believe that Rick Hugener is actually correct in terms of their model of consolidation? So let's go back. We've sort of stopped at LTP. We looked at why we study LTP the way that we do, and we study LTP the way that we do because Eric Kandel set up the model for us. We look at circuits, we look at simplified organisms, and we look at circuits. We look at strengthening of specific parts of this circuit, and then we try to find out if kinases will change some of the pre-existing proteins. And that's LTP, but that's not long-term memory. For that short-term LTP to become consolidated as a memory that you're going to be able to keep with you beyond sort of 1 day to a week to a month to a year to longer periods of time, we need to look at how does this memory leave the hippocampus to the fimbriae fornix, and how does this memory get stored long-term outside of the hippocampus in other areas of the brain in terms of the mirror of these synapses, which we call an engram or a memory trace. And this consolidation, we think, where we convert these short-term memories at the level of the hippocampus into long-term memories that can last years or maybe even decades, is something that we're not entirely sure of, but people are so incredibly interested in this particular field, and this is where all of this controversy comes into play. So we know, for example, if we're looking at how this process might look, we've been looking at what happens here. We've been looking at the strengthening of individual synapses. When you're trying to learn about all these different things within HMB300 or in all of your other courses, it's that short-term memory that you're trying to store things at the end of the lecture. If you remember anything from the end of this 1-hour lecture, that's stored in this short-term memory in these individual synapses that are now stronger with AMPA receptors. We have more activation of CAM kinase 2 at the level of the CA1 within the hippocampus. But you're also trying to store this in long-term memory, beyond sort of this 1 day, maybe past this week, maybe past this month, into long-term memory. If you remember this 1 year from now, you've been successful because it's left through that fimbriae fornix and is now sort of in the larger regions of the cortex, where certain synapses within the area of the frontal cortex have been mirrored and are also strengthened. I keep mentioning mirroring, and I'm going to show you this on the next diagram as well. At some point, the memories that you're trying to store up right now, the strengthened synapses, all of those individual synapses as you're trying to remember what is this fimbriae fornix, where is it on this particular slide, those AMPA receptors on those particular synapses are going to be associated no longer with the hippocampus. They're going to have left somehow and are now going to be associated with the cortex. We know that this process of storing memory long-term outside of the hippocampus, this consolidation of memory, where it is now being stored outside of the hippocampus, is protein synthesis dependent, because if we use protein synthesis inhibitors, things like actinomycin D or cyclohexamide, that these protein synthesis inhibitors, when we are blocking long-term memory, if we inject these into the brain, you will block long-term forms of memory. LTP is maintained. There are strengthened synapses. We can measure LTP, but there is no long-term form of memory. We also know that this requires CAM kinase 2, and this consolidation requires CAM kinase 2, likely because it involves LTP. Without LTP, we can't get consolidation, but it probably also involves CAM kinase 4. So here's where we are at right now. And this is what I want you to get across. For you to remember something long-term, and this is the work of individuals like Alcino Silva. So we looked at Alcino Silva in terms of the CAM kinase 2 mutant. And this is also Paul Franklin's work and Morris Moskovich's work, and they are here in Toronto. And what they say is that the short-term memory, we get LTP at individual synapses within the hippocampus. And these hippocampal synapses, as they get strengthened, will send out signals, and they will in turn make synaptic connections via their axons that leave as the fimbriae fornix in various areas of the cortex. So we have strengthening here, and these strengthened synapses in turn will send out their projections out into different areas of the cortex, and these cortical modules will also be strengthened as we try to repetitively keep that in this memory. Eventually, with time, the hippocampus, because we know long-term memories do not require the hippocampus. We know that from HM. HM had all of his childhood memories intact, but the hippocampus is no longer required for those memories because they are now stored in terms of these cortical strengthened modules that you see here. And over time, these strengthened cortical modules will retain all of the different properties of the strengthened synapses that originally existed within the hippocampus. So they are being mirrored. The synapses that were originally in the hippocampus are now mirrored, or there is a trace of these memories within these particular cortical modules that, again, over time, no longer requires input coming in from the hippocampus. It's a theory, and this idea that the cortical modules that you see here, certain cortical neurons are getting strengthened, and consolidation of this memory or these individual synapses is an important part of what we think happens within neuroscience. So it's believed that the hippocampus originally activates these groups of neurons within the cortex when it starts retrieving a memory. We don't really understand how this all works, to be quite honest. We think that the hippocampus is required for a while, but then it's no longer required. And I sort of left off with this, that the entorhinal cortex, for example, might be equally important. It's not the hippocampus that's important, and again, it's really controversial. And we know that the entorhinal cortex being damaged in Alzheimer's disease leads to the loss of very, very long-term memories. So whether it's the cortex, whether it's the entorhinal cortex, we're still sort of up in the air in terms of where long-term memory actually exists. So it's actually been about a month since I wore a unicorn costume. I don't know if you have that memory in your cortex or not, but you can imagine right now, if you do happen to remember that, if you were in class and you happen to remember that I wore a unicorn costume to class, that groups of neurons right now within your cortex are being stimulated to retrieve that memory at these individual synapses that exist within your cortex, no longer within your hippocampus, that memory exists because some of these synapses are being reactivated again. What makes those synapses special? You have so many of these synapses. Which of those synapses are the ones that are going to be activated by only the memory of those unicorns? And this is sort of where we get into this idea that memory consolidation involves synaptic tagging. Not all of your synapses are going to be involved or are all that important in remembering me dressed up in a unicorn costume. And in fact, it'd be great if I could figure out a way to identify each of those different synapses so I could completely erase that from your memory, to be quite honest. But how did that all come to pass? And one of these models involves this synaptic tagging model to develop a consolidated memory that exists within your cortex. Somehow, within your cortex, as information was being transferred from strengthened LTP synapses within the hippocampus, leaving the hippocampus through the fimbriofornical structures, their axons. One of the things that happens, some of those synapses within your cortex related to particular memories will also start strengthening within the cortex. And as they do so, there is some sort of signal that is being generated at those particular cortical synapses from the hippocampus through those fimbriofornical connections that leads to a memory tag being generated. And this is that model of a synaptic tag. So this memory formation requires LTP here as well. The same synaptic strengthening that occurred within the hippocampus initially now has to be mirrored, and we need LTP occurring at the level of the cortex. But in addition to the LTP, in addition to the CAM kinase 2 dependent mechanisms, the synaptic tagging for memory consolidation, for you to remember something longer term at the level of the cortex, requires that protein synthesis has to occur, because we can block all of this, again, using actinomycin D and cyclohexamide. So what is this synaptic tag molecule? What is this mystery molecule that's shown here that allows you to remember a particular thing or a particular event only at these synapses, but not at every single synapse? How do these synapses here remember event A, and how do these synapses in purple remember that I wore a unicorn costume? And how does it remember, and how do we retrieve this memory over long periods of time? Because these are getting strengthened as well, and what is being captured and what is being produced at these strengthened synapses? So this is sort of the search that many individuals, after Kandel and others, had figured out that LTP occurs within the aplysia, and LTP occurs within the hippocampus. How does consolidation lead to all of these events, not at the level of the hippocampus, but at the level of the cortex? And this search for these candidate molecules was an incredibly competitive one. Just like the 1950s and the 1960s with Holger Heiden and Egehazy and eventually James McConnell, people wanted to identify these molecules that were involved in consolidation. It had to be protein synthesis dependent, therefore new proteins had to be synthesized. What could this potential molecule be? So this current theory right now is that a memory is actually a network of these synapses, so these synapses that you see here, that are in a discrete group of neurons within your cortex. So a memory for a particular event, for example, only a certain group of neurons store that memory, and only a certain subset of the synapses within those neurons are storing that memory. And this network of neurons and their synapses generate what is known as this memory trace, which is also known as an engram. So only specific memories are related to a strengthening of these specific groups of these synapses. So if we can alter this memory trace or this group of synapses, we might be able to alter this memory. And in fact, recent work by Susumu Tanagawa and others at MIT have shown exactly that. So this race to understand what these memory molecules are that allow for this strengthening within the cortex was a really important one. And there was really exciting work done early in the 2000s to actually suggest that maybe people had actually figured out what this consolidation molecule was. And one of these things that was important is that it actually tied together a number of these different types of things that were going on at the level of the hippocampus, but potentially and more excitingly at the level of the cortex. So LTP is probably a great way for an animal to learn short-term, a different type of learning behavior that can eventually be stored in memory. But in terms of long-term memory, LTP is not enough to account for this. So we need strengthening of synapses, and we need presynaptic and postsynaptic to pair together. We need, again, at least at the level of the excitatory glutamatergic synapses, we need the release of glutamate. We need it to activate NMDA receptors. We know that we get a strengthening of AMPA receptors. But within the cortex, how does the cortex know that this synapse is one that is responsible for a particular memory and that the synapse right beside it is responsible for a completely different one? We need to have that specificity of the presynaptic synapses encoding for very, very specific forms of memory. So one of the most exciting types of research that came out was that if you were to take a look at molecules like RNA, particularly messenger RNA, within the axon and within the dendrites, we have a lot of mRNA. What that mRNA was doing was actually unknown for a very, very long period of time. People had described it, and people knew that mRNA existed, but it didn't make a lot of sense to neurobiologists for a very long period of time, because why would you want to have mRNA there? And this exciting theory that came out of Duden and Todd Sachter's group actually suggested that when you have late or very long-term forms of LTP, that the nucleus actually will create and transcribe PKM zeta, protein kinase M, zeta isoforms, and the mRNA exists at different synapses within the hippocampus, but also within the cortex. And that this PKM zeta mRNA can very, very rapidly, on the order of microseconds, as soon as this synapse gets activated, can actually be transcribed and translated very quickly to the level of a protein that can then be involved in strengthening of this synapse. So imagine that I'm trying to remember something, and this synapse is activated, and that this protein kinase A allows for me to remember because it is the memory molecule that's sitting underneath this particular activated set of synapses. We also know from Todd Sachter's work that if he used what is known as a ZIP inhibitory protein, so this is PKN zeta inhibitory peptide, and he were to apply this to the synapse, in fact, the potentiation, the strengthening, as well as the memory for whatever had been learned would be completely ameliorated or erased after its application. So we get LTP, and we look 24 hours later, so theoretically, beyond sort of the window of long-term memory, LTP alone, we're looking at LTM, or long-term memory. We apply ZIP, the PKM zeta inhibitory peptide, and as soon as we block PKM zeta from being transcribed and translated at the synapse, we immediately lose that strengthening. Again, we can look much later in terms of the behavior itself. The behavioral performance is also completely erased. So Todd Sachter and Jan Duden figured out that maybe PKM zeta is this molecule and this enzyme that's particularly important in this process. And they used a pharmacological inhibitor known as BZIP, or zeta inhibitory peptide, to specifically erase memory. Again, it was remarkable if you think a little bit about it. And again, I'm going to take you through all of these different types of things in the next slide, but I'm also going to show you a movie, if I can, to demonstrate what they were able to do. So a very simple type of learning and memory where an animal is going to learn by using visual cues that are available to it to avoid getting into the shock zone. And as long as it learns this and it stores it in long-term memory, so beyond 24 hours would be considered long-term memory, that it will avoid being in this zone here where it will receive a shock. So with the application of beta zip, which is down here, the animal, after it has been trained, will actually stay in that area, and so it has forgotten, or the memory of the training that has occurred has actually been erased. And I'm going to show you that on that next slide. So I'm going to hope that this video plays. So apologies, but it didn't actually seem to work, so I'm just going to take you through this. And one of the things that we see is that there is a rotating shock sector, and during the pre-training phase where there's no shock being delivered, the animal will be able to completely wander around, and this platform actually rotates, and you can see that these dots in between here actually indicate that the animal spends some time here. This is in the pre-training phase, and as it learns that it's going to get a shock, it learns to avoid this area, and in fact, it spends most of the time as the platform rotates moving out of that zone as much as possible. When you go back 24 hours after the last training has occurred, so again, long-term memory, hippocampal dependent because it's using these cues to know when it is entering into that shock zone, you will see that if it's only being injected with saline, that in fact it actually avoids getting into this shock zone quite a bit. Again, with the beta zip application, the animal does exactly the same thing. It learns exactly in the same way, so it avoids getting into this area. But as soon as the training is done, and now it is being injected with ZIP, which blocks PKM zeta, one of the things that we see is that we get the animal going back into this zone. Very, very different than what we saw here. So what this seems to just suggest is that because it is now spending more time within the shock zone, that this is the time that it would spend before training, that after it has learned this particular task, that it actually learns to avoid it, so it spends less time in the shock zone, and that with ZIP application, actually it has completely forgotten to avoid this particular area. Again, remarkable results if it turns out to be true. So how might all of this work? And why is this really all that exciting? So one of the things that I need you to be aware of, PKM-zeta is an atypical type of protein kinase. Protein kinases are things that people are interested in, mainly because of the work of Eric Kandel. And one of the things that we know is that protein kinase, Z, is actually a derivative of protein kinase C. So protein kinase C and the Z isoform of protein kinase C, the gene itself, has both a regulatory domain as well as a catalytic domain. And protein kinase M, which is derived from this, only transcribes the catalytic domain. The regulatory domain is not present. So the mRNA for protein kinase, PKM-zeta no longer has the regulatory domain. So when this is actually translated, it becomes fully activated, and there's no way to actually turn this off. And again, at the level of the synapse, protein kinase M-zeta may actually assist in reactivating some of those AMPA receptors, which again makes some sense in terms of what might be going on. So again, we don't know how exactly this all occurs, but we know that this might be microtubule dependent, that the trafficking from the nucleus all the way out to the dendrites might involve microtubules, things like tubulin. It might involve transport molecules like kinesin family member 5 or KIF5 to transport this anterograde out toward the different spines within the different dendrites. So again, there's a lot of other work that also seems to implicate that PKM-zeta is important in long-term forms of memory. And Sackter's group, again, since their publication in the early 2000s, back in 2006, also generated a great deal of interest. They had published a number of other papers, one of which I'm going to show you next, but they also faced a lot of criticism. So some of this is that only their group, and this is a major criticism, only their group was able to show that PKM-zeta was involved in long-term memory. Only their group was able to show that ZIP application completely erased memory that involved PKM-zeta. The other major caveat to all of this involved the fact that PKM-zeta was still being applied at the level of the hippocampus. Remember our earlier discussion where we were looking at PKM-zeta and we were looking at memory molecules? What I had shown you earlier in that consolidation model is that the cortex is important in long-term consolidation of memory traces. And we're still at the level of the hippocampus. We're still thinking about how the hippocampus is being affected. And so this didn't really fit well with the model of cortex. It didn't really fit well with the cortex being strengthened, and their group is the only one that is able to do all of these different types of experiments. In addition to that, 24 hours, even though they consider this a form of long-term memory, actually isn't long-term at all. So they faced all of these different criticisms, and they came up with another high-profile paper to actually address some of these different issues. So one of the things that I need you to make sure that you know, especially for the quiz as well as for other things that are coming up, is this conditioned taste aversion task that is going to be modeled for you. So in this particular type of task, the rat will undergo water deprivation, which is what you see here as WD1. So water deprivation over a series of 3 days, so they get very, very minimal amounts of water, so they become very, very thirsty. And then one of the things that happens is that they will then be allowed to drink water, one that is saccharin flavored, so this is sweet tasting water, or just water alone. And again, the animal normally prefers getting the sugared or flavored water, the saccharin flavored water versus water alone. So we can see if, for example, it will want to take up more of the saccharin-flavored water. However, in this conditioning phase that you see here in the middle, one of the things that you can go in and do at the same time that you are allowing them to drink this water is to also inject them with lithium chloride. And lithium chloride injection at the same time that they start drinking this water makes an association for them. And this association is that this lithium chloride makes them very, very nauseous. So they throw up and they get very sick. So every time they go in and they drink flavored water, they remember being sick. So that is a form of learning that does not involve the hippocampus. So it is hippocampus independent. And this conditioned taste aversion task is very, very long term. So you might remember, for example, something that might have happened to you a long time ago, where, for example, you might have eaten something that made you sick, and you will avoid eating that for a very long period of time because you don't exactly remember all of the context around it, but you remember it did make you sick and it made you want to throw up. And so when you go back and look and to see if you have a preference for sucrose versus like water, now under experimental conditions, the animal will actually no longer prefer having sucrose or saccharin flavored water. It actually prefers just regular water. Now, why is this all important for Todd Sachter and his group? One of the things that we know is that this is a very different form of learning that does not involve the hippocampus. So ding, good for Todd Sachter. Also, this is a very long-lasting memory. We can go back in a month later and go back and test. It's not one day later, it's months later. So this truly is long-term memory. We also know that this involves the cortex. So how long can we go? In fact, if you were to go in and look at the aversion index, you can actually see that compared to the vehicle, that the animals will actually avoid drinking saccharin if it was injected with lithium chloride up to 25 days. And they will avoid that and have this conditioned taste aversion for up to 25 days. They will prefer drinking the water versus the saccharin flavored water. However, if you were to go in and you were also to inject them with ZIP, and you were to go in after they had just learned this task, so that you do that training, and you do it one day later, you inject them with ZIP. In fact, even though they had been trained and they got sick, and they should normally have that taste aversion, that with ZIP application, in fact, they have completely forgotten that sucrose flavored water or saccharin flavored water, they should be avoiding it. And 3 days after they were injected, or 7 days, or even 1 month later, they are now no longer avoiding that saccharin flavored water, and they have forgotten that conditioned taste aversion. Again, another reason why this is all really important is that when you look at the areas of the brain that seem to be important, the areas that seem to be activated in humans are the basolateral amygdala, but more importantly, the insula. And the insula in rats, as well as in humans, where we have a strong aversion or when we have a sense of disgust, is really where we integrate with the limbic area of the brain in the cortex. So this stored form of memory, the consolidation of this memory, the strengthening of synapses is not in the hippocampus. This is actually within the cortex. So the insula, which is shown here and as well here, is actually really important in terms of long-term memory related to aversively motivated tasks, just as we saw earlier with the conditioned taste aversion. So all of this again suggests that Todd Sachter's group, by injecting ZIP into these areas, were directly affecting the strengthening of synapses, the consolidation of memory, and that this involved the erasure of memory by blocking this PKM zeta. So this is now in the cortex, not the hippocampus. So ding, good for Todd Sachter. This is also very long term, 25 days later. So also ding for Todd Sachter. And this also showed a proof of concept that it's outside of the hippocampus, and his zip is also working here as well. So I'm going to actually stop here, and hopefully you'll have the opportunity to go back and review this material. Much of this will be on your upcoming quiz. Please spend time reviewing as much as you can. We'll go back to the normal regular lecture schedule in the upcoming weeks. We will also try to make sure that all of this material is available as different forms of streams for you as well. And we will also be posting an announcement soon about the upcoming review session that we have planned for December the 14th, the Friday before you actually write your term test. So I want to make sure that all of you are doing well in terms of your mental health. Please stay strong, get lots of rest, make sure that your consolidation of your memory is going well, and please let me know if there are other things that I can provide for you in terms of your studying and to make sure that your stress levels are manageable. We do have plans for you also in terms of the study guides. A group of students will be going back to the original format of the study guides, and they should also be available for you for this lecture in the next little while. So thank you. Have a wonderful day. I hope to see you back in class. My own anxiety is now sort of more or less under control, and I'm hoping that we can go back to our normal lecture schedule in the upcoming week. Thank you so much for understanding. I greatly appreciate it. I do miss seeing all of you in person, and I'm hoping to have my anxiety under control by next week. Thank you very much. Have a great weekend, and I look forward to seeing you next class.